So let's examine some cases here. Let's suppose that we look at here at our gene. We've got our promoter, PLAC. We've got the operator site here. We've got the LAC-Z gene here. And we've got the LAC repressor, LAC-I, the repressor sitting there. Polymerase tries to come along to this, and polymerase tries to come along to this, and it's blocked. So what will happen in terms of the transcription of the LAC operon? No mRNA. So that's great. So we've solved one problem right off the bat. We want to be sure that sometimes there's going to be no mRNA made. This way, we're not going to waste any metabolic energy making beta-galactosidase. Are we done? No. Why not? We got to sometimes make beta-galactosidase. So we got to get that repressor off there. Well, how's the repressor going to come off there? When do we want the repressor off there? When there's lactose present. So somehow we need to build some kind of an elaborate sensory mechanism that is able to tell when lactose is present and send a signal to the repressor protein saying, hey, lactose is around. We sense that. The signal gets transmitted all the way to the repressor protein, and the repressor protein comes off. What kind of an elaborate sensory mechanism might we build? Use lactose as what? So you're saying, like, so this is actually pretty simple. You're saying just take lactose, and if lactose, you want lactose to be its own signal? So if lactose were to just bind to the repressor might then know that there was lactose around. Well, what would it do if lactose bound to it? Sorry? Fall off. Why would it fall off?
Well, okay, so if it's bound to the mRNA,
yep.
and then lactose comes along and binds to it, it will be like more interested in the lactose, so it'll bind to the lactose.
More interested in the lactose. So if your suggestion, this is good, I like the design work going on here. The suggestion is that if lactose binds to this here, binds to our repressor, it's going to fall off because it's more interested in lactose than in the DNA. Now, how is the interest actually conveyed into something, material? Because the actual level of cognitive like or dislike for DNA on the part of this amino polypeptide is unclear. We may be anthropomorphizing slightly with regard to this polypeptide chain. So mechanistically, what's going to happen? Shape. Yes, shape? Change conformation. The binding act, the act of binding lactose creates some energy, may change the shape of the protein, and that shape of the protein may, in the process of wiggling around to bind lactose, may de-wiggle some other part of it that now no longer binds so well to DNA. That is exactly what happens. Good job. So you guys have designed, in fact, what really happens. What happens is what's called an allosteric change. Just means other shape. So it just changes its shape. That it changes shape on binding of lactose. And it falls off because it's less suitable for binding this particular DNA sequence when it's bound to lactose there. So, in this case, in the presence of lactose, LAC-I does not bind, and the LAC operon is transcribed. Yes? Uh-oh. Okay. All right, designers, here we got a problem. You have such a cool system, right? You were going to sense lactose. Lactose was going to bind to the lac repressor, change its conformation, fall off. Uh-oh. But as you point out, how's it going to get any lactose? Because there's not a lactose permease, because the lactose permease is made by the same operon. Oh, so what if, in fact, instead of getting one of these DOD mil-spec kind of things of some repressor that is absolutely so tight that it never falls off under any circumstances, what if we build a slightly sloppy repressor that occasionally falls off and occasionally allows transcription of the lac operon? Then we'll have some trace quantities of permease around. With a little bit of permease around, a little lactose will get in. And as long as even a little lactose gets in, it'll now shift the equilibrium so that the repressor is off more. And of course, that'll make more permease and shift and shift and shift and shift. So as long as it's not so perfectly engineered as to have nothing being transcribed, so no mRNA is really very little mRNA. See, this is what's so good, I think, about having MIT students learn this stuff, because there are all sorts of wonderful design principles here about how you build systems. And I think this is just a very good example of how you build a system like this. Now, all right, so we now have the ability to have lac on and lac off, and that is lac off, mostly off, because of your permease problem. Glucose control. So here's my gene. Here's my promoter, P-lac. Here's my operator, beta-gal is encoded by lac-Z. We've got all that. When this guy is present, when lactose is present, the repressor comes off. Polymerase sits down. Wait a second. Polymerase isn't supposed to sit down unless there's no glucose. We need another sensor to tell if there's glucose or if there's low glucose. So we're going to need a sensor that tells that. Any ideas? Yep. Yeah, if you work that one through, I don't think it quite works, but you've got the basic idea. You're going to want another something, and it turns out there's another site over here. Okay? There's a second site on which a completely different protein binds, and this protein is the cyclic AMP regulatory protein. And it so happens that in the cell, when there's low amounts of glucose, make sure I've got this right, when there is low amounts of glucose, what we have is high amounts of cyclic AMP. Cyclic AMP turns out, whereas lactose is used directly as the signal, cyclic AMP is used as the signal here. When the cell has low amounts of glucose, it has high amounts of cyclic AMP. Now, what do you want your cyclic AMP to do? How are we going to design this? It's going to bind to a protein, the cyclic AMP regulatory protein. It's going to sit down. And now what's it going to do? Is it going to block RNA polymerase? What do we want to do if there's low glucose, high cyclic AMP, we sit down at the site, we want to turn on transcription now, right? So what it's got to do is not block RNA polymerase, but help RNA polymerase. So what it actually does is instead of being a repressor, it's an activator. And what it does is it makes it more attractive for RNA polymerase to bind. And it actually does that by actually does it slightly by bending the DNA, but what it does is it makes it easier for RNA polymerase to bind. It turns out that the promoter is kind of a crummy promoter. It's actually just like, remember, the repressor wasn't perfect, the promoter's not perfect either. The promoter's kind of crummy. And unless RNA polymerase gets a little help from this other regulatory protein, it doesn't work. We have two controls: a negative regulator responding to an environmental cue, a positive activator responding to an environmental cue, helping polymerase decide whether to transcribe or not. And basically, that's how a human egg goes to a complete adult and lives its entire life, minus a few other details.